My name is Tanja Hyvärinen and I am PhD student in the group of adjunct professor Susanna Narkilahti at the Faculty of Medicine and Health Technology at Tampere University. Next I will present our recently published work on differentiation of human pluripotent stem cell-derived cortical neurons on laminin isoform 521. Major focus was on the development of functional activity in these stem cell-derived cortical networks and how they compare with the widely used rat cortical cultures. Human pluripotent stem cells, hPSCs, have the capacity to self-renew and differentiate into all cell types of the body. hPSCs include the human embryonic stem cells as well as the human-induced pluripotent stem cells, which are reprogrammed from the adult somatic cells. Stem cells have great potential in neuroscience applications, including disease modeling and drug development. Multiple protocols have been developed for the differentiation of cortical neurons, all of which aim at the fast and consistent production of neurons. During in vitro differentiation, the precursor cells are first formed through neural induction and specification. These neural precursor cells can be further differentiated into mature cortical neurons. However, generating functionally active networks has been a challenge in the field. The rationale behind this study is based on specific properties of laminin molecules. Laminins are a major family of extracellular matrix molecules that are essential during both embryogenesis and neural development. Several earlier publications have shown that laminin isoform 521 is a suitable substrate for the maintenance of human pluripotent stem cells. Our group has also previously presented that the laminin alpha-5 substrates in particular improve the survival and functionality of stem cell-derived neurons. Therefore, LN521 was chosen as the sole substrate for maintaining the pluripotent stem cells and for supporting neural differentiation that is presented in more detail by the schematics. Use of these defined substrates instead of animal-derived products can increase the experimental reproducibility. Furthermore, laminin alpha-5 substrates are shown to promote the functional development of neurons. Neural induction on LN521 substrate was efficient as shown here by the appearance of PAX6 and vimentin-positive neural precursor cells. To test repeatability of neural induction, we performed experiments with 3 pluripotent stem cell lines from both embryonic and induced pluripotent stem cell origin. In all cases, we saw efficient neural conversion with over 90% PAX6 positive cells after 12 days. The number of precursor cells gradually decreased in all cell lines with neuron maturation. With further maturation, these precursor cells differentiated into neural cell types, MAP2 and beta-tubulin positive neurons, and S100 beta and GFAP positive astrocytes. We could also identify neurons expressing different cortical layer specific markers, shown in the right figure panel in green. Functionality of neurons can be measured in vitro on multiple levels, either from single cells or at the network level. The network level activity can be measured, for example, utilizing microelectrode array technology, the MEAs. MEAs consist of a grid of microelectrodes at the bottom of the culture well, which measure neuronal signals arising from different regions of the network. The method is non-invasive and allows repeated measurements over weeks. Our group has published first MEA experiments with stem cell-derived neurons already 10 years ago, and have since utilized this method in many projects. First, we showed that LN521 is indeed superior in promoting functional activity as compared to the more commonly used mouse laminin substrate. On the left, you can see 6 individual cultures of both mouse laminin and LN521 differentiated neurons on MEA. The heat map is showing the rate of detect activity. To evaluate the development and patterns of activity, we compared these human cortical networks with rat embryonic cortical cultures, which are really considered as the golden standard, especially in the field of in vitro MEA studies. The movies are from a single MEA well with 64 electrodes, and the recordings from hPSC and rat networks reveal organization of activity into highly synchronous network events. Importantly, both networks reach similar level of activity, and the functionality develops in a comparable time period. Functional connectivity is typically measured as simultaneously occurring activity in different areas of the network. On the right, we have connectivity maps showing functional connections on MEA over several measurement days. Quantification confirms that both cell types formed highly connected networks, typical for cortical neurons. Principal component analysis can efficiently cluster MEA data based on activity feature profiles. We calculated several different parameters from the activity patterns and applied PCA to compare activity features of hPSC and rat networks. We saw clear segregation of hPSC-derived and rat recordings into separate clusters, which is likely to reflect the species-specific and maturation state differences between these cell types. As a summary, hPSC-derived cortical networks were generated using highly defined LN521 substrate, resulting in consistency of differentiation. The development of functional activity was fast, and the time frame similar to the rat cortical cultures. We could see formation of highly connected networks, typical for cortical type activity. However, the hPSC neurons displayed distinct activity patterns from rodent counterparts. And finally, these functional hPSC-derived cortical networks can serve as a human-specific model for neuroscience applications, for instance, pharmacological studies and modeling network dysfunctions. If you want to read more, take a look at our recent article in Scientific Reports. I would also like to acknowledge our funding support, my supervisor, all co-authors, and the following people who have contributed to this work. If you have further questions, here is a link to our research group web pages and to my LinkedIn profile. Thank you.